I'm going to try to stick with the facts here. So I brought my notes so that I can be perfectly accurate. As you've seen in the newsletter, Aubrey is a PhD in biology. He has his PhD from Cambridge University, which of course is tremendously wonderful. He is author of 47 peer-reviewed articles, some of which he reviewed himself, because he's the editor-in-chief of the journal Rejuvenation Research. He is the co-founder of the SENS Foundation. SENS, S-E-N-S, stands for Strategies for Engineered Negligible Senescence. Senescence is another fancy word for aging. And it's quite a mouthful. I was curious how he came up with that name, and I'm told that he is someone who has always wanted to make sense. Now, all of the above information, one other thing, he's also the author of a very excellent book, which is selling very well, called Ending Aging. Now, all this information comes right out of the newsletter, but we also, of course, do our due diligence in the Smart Life Forum, because we are concerned to screen out people who are on the payroll of Big Pharma, and we don't want to, we oppose manipulative marketing, and so we wanted to make sure that he was on the up and up. And so, and we did that, and found out he was an honest fellow, committed to the public good, but we also found out a couple of other things. By the way, I use the same private investigator that Mike Corrick uses. And a couple of things that people don't generally know about Rudy Gray. The first one is that he's in secret negotiations right now with the Vatican. And that, you can imagine how that happened, because, as you know, the Vatican is concerned about, well, they're concerned that his book should be placed on the index of prohibited books. Their position has always been that birth control violates the natural law and is unnatural. And similarly, that death control, there's an argument being made by some people in the Vatican that death control is also contrary to natural law, and that therefore books which advocate it should be placed on the index. Now he's arguing, I think quite persuasively, that the Church's pro-life can't be opposed to unnecessary death, and secondly, that if indeed death control is unnatural, then all medicine practiced to save lives is unnatural. And so there's hope that the Church will abstain from taking that drastic action. Now the other thing is even more remarkable. It turns out that, Aubrey de Grey, being a biologist, is a student, or was a student, of the famous Gregor Mendel. Now you may know about Gregor Mendel, a man who studied peas and is kind of, in a way, the father of genetics. And Gregor Mendel was a monk who, his order was established in his facility, well, I guess the monastery, was in Austria. Very interesting, he studied peas, and he looked at the parent plant, and then the offspring plant, and then he would see how the traits were passed on, and how the, whoops, wait a minute, what are you doing? Okay. And how the traits of the parent were passed on to the, in a sense, inherited by the descendants. So, as a matter of fact, the Mendel called the parent plants peas, and the descendants he called cues. Cues from quintuplets, because in the pea pod there are 5 peas, and so the descendants were called quintuplets. And so, therefore, his
we can hear you better if you move that thing from in front of your mouth.
Yeah. No, it's got a short in it or something.
Dead battery.
Okay.
Working with this.
It's got a dead battery.
So anyway, his brothers became concerned because they didn't think he was praying enough. He was spending too much time minding his P's and Q's. Now, this expression it was picked up by people studying the propositional calculus, as you know. So he's a very influential man. Now the interesting thing, though, the relevant thing that relates to Aubrey de Grey is that Gregor Mendel published most of his work in the 1860s, and he died in 1884. That means that since Aubrey de Grey was his student, that he must be probably 130 years old. Now this is not surprising when you meet him. Thank you. Should I use it? Sure. Okay. And because his wonderful beard makes a strong impression, and so
we can hear you good without it.
okay, set it down. When you speak with him, you find out that he's a very young man. And so he's a very young man with a very old beard. Now, in fact, here's the fascinating thing. He is the first young man to be over 100 years old. And in fact, I think he is the oldest young man that we are likely to meet anytime soon. Aubrey.
No one told me about these introductions. All right. So thank you very much to everybody for having me. It's a great pleasure to be here. It's always a great pleasure to speak to a new audience, and especially one that has such a long-standing interest in the sort of thing I do. Now, I was particularly fascinated with the erudition of Steve's talk earlier on. I didn't really know that there were people here who were quite so knowledgeable about supplementation and other stuff like that. And luckily, I'm not going to have to match it, because I'm not going to be talking about supplements at all, as you'll shortly see. I'm going to be talking about something which is perhaps complementary to that. First of all, I'm going to talk a little bit about the organized... Yeah, that's true. Yeah, lights. Going there? That's the wrong... I think you turned off the wrong lights. Result. Okay, good. Yeah, so this is my title, in case none of you could see it earlier on. And that's my email address and the website of my foundation. And I'm always happy to be emailed and to try to be responsive if you have any questions later on. So those of you who have heard of me before will probably think that I work for an organization called the Methuselah Foundation, which indeed I did work for from about 2002, although at that time I was only working for it in my spare time, through until about 2 months ago. But the Methuselah Foundation has divested its research funding activities to a new foundation called Sense Foundation. We did this for a whole bunch of reasons to do with messaging and fundraising and tedious stuff like that. But the Sense Foundation is now the charity that I am the Chief Science Officer of, and that orchestrates the promotion of the sort of work I'm going to be telling you about this evening. As you see, it's a US registered charity, and we work to develop and promote regenerative medicine solutions to the age-related ill health that we're also familiar with. We have, over the past year, another thing that some of you may not know if you thought you knew what I do, we have now an in-house laboratory. Historically, we did the funding of research purely as a funding body. In other words, we gave money to people in universities around the world to get research done. But over the past year, we have built up a small but rapidly growing and extremely efficient in-house laboratory in Arizona, which we are hoping to move actually to this general area of the country fairly soon. And of course, we do a lot of outreach. I give the most insane number of talks to audiences of all different types, including you. So that's why I'm here. So what I'm going to do is I'm going to talk for probably 3/4 of an hour, and I'm going to split my remarks into these 6 areas. First of all, I'm going to tell you in very broad, quite abstract terms really, why I think that there is a new way that we might be able to go about really combating aging. And I've got to tell you what that way is. Then I'm going to go in the second part of the talk into a little bit more detail. I'm going to describe how I break that down into 7 major sub-problems, if you like, and the solutions to them. Now, as you will see by that time, it's complicated. I want to do an awful lot of things to the body, and in order to explain in detail what I want to do, I think I would need a great deal longer than an hour. So I'm going to spend just a short amount of the talk just going into a lot more detail, a bit more detail anyway, about just one of those 7 areas. Then after that, I'm going to be talking about the longer term. I'm going to be talking about what I think is going to happen maybe 30 or so years from now and beyond with regard to the benefits for human age-related health and longevity that people, perhaps young people alive today, maybe in fact people who are in middle age today, may expect to be able to enjoy. And finally, I'm going to end with a little bit about how you folks can make a difference. Okay, so I'm going to start there. And I'm going to start off by giving you my favoured definition of aging. Those of you who are in the audience today, since you're very interested in this topic, probably have a pretty good idea of your own of how you would define aging. I like to define it this way. Clearly we know that aging is a side effect of being alive in the first place. In other words, metabolism, which is the word I'm going to use to encompass all of the biochemical and cellular processes that go on from one day to the next to keep us alive, has side effects, byproducts and so on, that build up and eventually cause age-related ill health, which I'm going to use the word pathology to denote. But what I'm describing here, as you can see, is I'm saying that in 2 stages. I'm going to talk about this word damage quite a lot this evening, and I'm going to use the word damage in this very precise way, as the set of intermediates between metabolism and pathology. So what I mean by that is damage is the set of molecular and cellular changes that happen in the body throughout life, even starting from before we're born, as side effects of metabolism, but which eventually cause age-related ill health. They cause pathology, or they contribute to pathology. And until that time, through most of life in other words, the damage is harmless. It's only harmful when there's too much of it. And you'll see why that's useful over the next few slides. What that means, classically, in terms of what we might be able to do about aging, can be summarized, obviously very, very briefly and concisely, in the diagram I'm showing here. We can either take what I'm going to call the gerontology approach, or we can take the geriatrics approach. Geriatrics approach says, well, okay, we've got this process whereby metabolism causes damage, causes pathology, and when the damage starts to build up and the pathology begins to emerge, we might be able to start attacking these pathologies and trying to effectively hold back the sands of time and slow down the rate at which these pathologies become life-threatening and really problematic. That's essentially the geriatrics approach. And a lot of what people do when they're interested in supplements and exercise and other optimizations of aging, so to speak, is essentially that approach. It's trying to slow down the progression of the pathologies that happen in old age. And there's nothing wrong with that. That's a fine idea. It will somewhat delay the age at which pathologies actually emerge. Exactly how much? Well, that's debatable. And of course it varies from one person to another. But not very much, not an enormous amount. Now, the gerontologists of the world know this, and they have said to themselves, well, prevention is better than cure, really, and it would be much better if we could dive in at an earlier point in the chain of events. If we could, instead of going in here, we could actually clean up metabolism. In other words, slow down the rate at which metabolism creates these various types of molecular and cellular damage in the first place. And the idea here is that we might be able to have much better bang for the buck. We might be able to actually have much more multifarious pleiotropic effects on the pathologies of old age with relatively simple interventions. And if we come back again to the benefits of supplementation and exercise and other relatively straightforward interventions that you guys are particularly interested in, then some of those come into that category as well, things that one might think would be sensible to take all one's life. However, I've already alluded to the fact that I don't really think that the geriatrics approach is going to be particularly beneficial. It's better than nothing, but not much better. This is basically why. I mean, you don't need to be a biologist to know that an awful lot of things go wrong with you as you get older. But of course, the main problem also is that because we're intervening rather late in the chain of events, the geriatrician's job is getting progressively harder as time goes on. Because these things I'm calling damage, in other words, the precursors of the various pathologies of aging, those things called damage are continuing to accumulate. So the geriatrician's job is getting harder. Unfortunately, the gerontology approach is actually also not terribly promising, or at least not yet, and this is essentially why. Metabolism is awfully complicated. This is, of course, a simplified diagram of a small subset of what we know about how cells work, and that's bad enough. But of course, what's really bad, as any biologist will tell you, is that this is a simplified diagram of a small subset of what we know about how cells work, which is completely dwarfed by the astronomical amount that we don't know. Even leaving aside all the stuff that we don't even know that we don't know. So basically, yeah, I mean, it's an extraordinarily complicated machine, the human body, and we understand it incredibly poorly. So the idea that we could actually significantly clean it up, significantly improve on what evolution has already laid down, is, you know, pretty far-fetched, really. We're going to do more harm than good if we do anything at all. Most of the time when we try to mess around with metabolism, metabolism laughs in our face and gets on with what it was doing anyway. Yeah, it's going to be hard not to do more harm than good. Now, of course, there are exceptions. And I wanted to say a few words about this, especially because this audience is quite sophisticated when it comes to what we can call magic bullets. In other words, relatively simple interventions, supplements, drugs that might benefit metabolism. I don't want you to go away tonight thinking that I feel that such interventions are completely pointless, because that's not what I think. What I think is that they have their place, but that it's equally important to make sure that you do not get overexcited about the amount that they can do. And a lot of gerontologists, it must be said, a lot of biologists who study aging make this mistake. So it's not surprising that a lot of lay people also make this mistake. There are a number of reasons to suppose that this is not really what we want to be looking at if we want to make a big amount of damage to aging. One problem is that if we look at short-lived species in the laboratory, the sort of species that have had their lifespans quite significantly extended using relatively simple techniques, you know, we might get the idea that we might be able to do the same for the human body, for the human species. But actually, there are good evolutionary arguments to suggest that the longer the natural lifespan of a given species, the less impact simple interventions are going to have. Essentially, the simple interventions will only work at all if the mechanisms that make them work are already in place, and it's really just a switch flipping exercise. And there are evolutionary reasons to suppose that we don't have such powerful switches in long-lived organisms like us. The next problem is that actually many of the things that have been shown to have significant effects in the laboratory are things that only seem to work if they are imposed throughout life, or at least starting quite early in life. If they're imposed late in life, then they have much less effect, if any. And this may very well be true for humans as well. There are some reason to believe that the increase in lifespan that we've seen over the past maybe 30, 50 years in the industrialized world, which certainly has been as a result of lower mortality rates in the elderly, has nevertheless actually arisen as a result of things that happened to those people very early in life. But the big problem is this. The big problem is that those of you who have the misfortune to be already going downhill a little bit would probably like to be put back together again, rather than to live longer in your somewhat diminished state. Of course, it's better than carrying on becoming more diminished, but you know, it's not ideal. So I'd like to look at a way of aiming a bit higher. And I'm going to start here. This car is about 50 years old. And of course, most cars don't last that long. Most cars maybe last 10 or 15 years before you junk them and get a new one. But there's a reason why this car lasted longer, and that is because it was built better. So this is the this is the vehicular equivalent of the human body. Humans obviously live unusually long compared to most mammals, most animals. This car lives unusually long, and that's because it was built well. But there's a reason for the particular title that I've given at the top of the slide. And the reason is, of course, that there are just as many 50-year-old VW Bugs driving around the streets of the world as there are 50-year-old Land Rovers. And it's not because the VW Bugs were built to last. The VW Bugs were built with something else that made them last a long time, namely style, which caused their owners to fall in love with them and do an exceptional amount of maintenance on them over the years, which is why this car, which was built in the same year as the one on the previous slide, is doing just as fine as the one on the previous slide. Now, what does this mean for the human body? What does it mean for aging? Well, it means something rather profound and rather easy to explain if I go back to my previous diagram. It shows you that there is actually a third, completely different approach that we could take to the postponement of aging. And I'm going to call it the maintenance approach. Sometimes I call it the engineering approach. Essentially what it is rather than coming in here and trying to clean up this process, trying to slow down the creation of damage, or going in down here and trying to slow down the creation of pathology, we go in the middle and we say, let's try and actually get rid of damage that's already been laid down beforehand. And this is a way of essentially uncoupling metabolism from pathology. We, on the one hand, are intervening early enough in the process that we don't have to worry about essentially the downward spiral, the vicious cycle of pathology that is the nemesis of the geriatrician. But also, at the same time, we're intervening late enough in the process that we don't actually have to understand metabolism. The idea here is that we let metabolism lay down these various types of damage at the rate that it naturally lays them down. And that means that we can just let metabolism get on with it, and of course that means we don't have to understand it particularly well. So what this comes down to, in my view, is that unlike these other two approaches, the engineering approach, the maintenance approach, does have a respectable chance of achieving a big extension of healthy human lifespan really rather soon. Now, that's my first outrageous claim of the evening. There's plenty more to come, don't worry. In order to get to the second one, I'm going to move on to the second part of the talk, which is to tell you really what this thing damage is that I've spoken about in this rather abstract manner so far. And in doing so, I'm going to justify the statement I'm going to make on this slide, which is that not only is the maintenance approach promising because it seems to target the weak link in aging, but also it's promising because that weak link is simpler than either its causes or its consequences. This is all we have to worry about when it comes to damage. Here I am listing 7 major categories of damage, 7 major categories of molecular and cellular phenomenon that are byproducts, side effects of metabolism that accumulate throughout life and that eventually contribute to age-related ill health. So things like indigestible molecules that accumulate in one or another way in the cell and because the cell doesn't know how to break them down. Or cells dying and not being automatically replaced by the division and differentiation of other cells. Or mutations in our 2 different genomes, and so on. Simple things. Now, the first thing I want to impress on you is that we have good reason to believe that this classification is complete, it's exhaustive, that there is no 8th thing lurking out there. And I can give a biological explanation for that, which essentially says, well, damage can only accumulate in long-lived structures, because if, let's say, a protein is synthesized and then it does its job and then it's damaged and then it's broken down, and that damage isn't accumulating. But also I can give a circumstantial but rather persuasive justification for my claim that this is an exhaustive list, which is this one. It's been the same list for more than a quarter of a century. We've come a long way in that time in biology and our ability to analyze biological systems and figure out what's going on with them. And you'd think we'd have added to this list by now if there were things to add. So that's pretty good news. But this is the really good news. At least I reckon we have a respectable shot at actually fixing these things. Of course, there are many more than 7 things that go wrong in aging. There are thousands of things, and this is just a way of classifying those things into 7 categories And one can classify 1,000 things into 7 categories in an awfully large number of different ways. So what's useful about this particular classification? The answer is that on this slide, you can see the same list on the left, and on the right, we can describe how we could go about actually fixing these things. Some of the fixes that I'm writing on the right-hand side are already very much part of contemporary, or at least imminent medicine that's in clinical trials now. So for example, cell loss, which I just mentioned, which is responsible for some aspects of aging, that's essentially the thing that is the target of stem cell therapy. That's what stem cell therapy is for, basically. Amyloid, something that happens in Alzheimer's disease, as I'm sure all of you know, and also in other organs during aging. The accumulation of extracellular proteinaceous aggregates, which is what amyloid is. There are clinical trials to get rid of that by using vaccination, by stimulating the immune system to engulf this stuff. Some of the things that we need to do, at least that I think we need to do, are a good deal harder than that, a good deal further away from the clinic. But in all cases, I think we have at least the ability to describe in quite a lot of detail what we need to do in order to get this to work. And that's why I think we can now talk about these therapies that we could implement well enough to give this amount of extra life, an extra 30 years or so, as being foreseeable. I think that we have a 50% chance of developing these therapies within as little as 20, 25, 30 years. Now, that may sound like a long time, and it could be quicker if we throw more money at the problem, I'm quite sure, but at least it means it's time to really start getting active and having a go, as opposed to pursuing a continuation of the exploratory approach. Well, it's certainly a good idea to carry on doing basic science on aging, figuring out more and more about how we age, but now is the time to throw a serious proportion of our effort at the exploitation of what we already know for the development of therapies that might really work. So I call this milestone robust human rejuvenation. And the key point about it is, of course, that it will be an additional 30 years of life given not to people who are born at the time that these therapies are developed, or who are in childhood, but who are already in middle age or perhaps older. So that's why it's relevant to people who are very much alive today. Now, so far, I seem to have made the whole thing sound rather simple. And of course, you all know that aging is not simple at all, and that if fixing it was simple, then we'd be a great deal closer to doing so than we actually are. So I want to spend the next few minutes reassuring you that I know that this is very hard. And in order to do so, I'm just going to go into one of the 7 different things here. I'm going to talk about this one, the accumulation of indigestible junk inside cells. And I'm going to talk about the way that we are trying in the Sense Foundation to go about addressing this. And this arises from a concept that I put forward about 10 years ago, which is attracting quite a bit of attention now. But there's still a long way to go, as you'll see, before we can really get it working. If someone could get me a glass of water, that would be terrific. Thanks very much. So what do I mean by intracellular junk? Well, here's an example. This is in Alzheimer's disease. This is an electron micrograph through an axon, in other words, the long part of a neuron. It's showing you that this axon is pretty unhappy. These things here are basically marvelous, thank you. Things called autophagosomes, which are supposed to be involved in the degradation of material inside the cell, and they're just basically not working. The things on the right are immunoelectron microscopy for various proteins that should be involved in breaking things down that have not succeeded in doing so. The same sort of thing happens elsewhere in the body. So this is in the artery, and as you can see here we have something that looks completely different, but it's fundamentally the same problem. You've got stuff accumulating inside the cell. In this case, it's oxidized cholesterol, and the cell is not working as well as it used to as a result. And this is the beginning of atherosclerosis. So what are we going to do about it? Well, the idea I had about 10 years ago was to apply to this problem a technology that had not previously been applied to anything biomedical at all, but rather a technology that comes from the field of environmental decontamination. Bioremediation is a concept that was first introduced about 50 years ago, and it's summarized on this slide. Essentially, the paradox that people wanted to understand was why, when you go and isolate microbes from any particular environment, you tend to find an awful lot of different species of microbe. The natural presumption would be that you'd only see one or two, because some microbes grow faster than others, and you'd expect the fast-growing ones just to outcompete the others. That doesn't seem to happen. And it was realized that the reason it doesn't happen is because if there is some substrate, some foodstuff in the environment that is hard to break down, hard to eat, then maybe only one or two of the species of bacteria in that environment will be able to eat it. And even if they're rather slow-growing ones, that will be good enough. That will give them their ecological niche, because their faster-growing competitors will not be competing for that particular food source. So it was realized that if you have got, let's say, a disused airfield, and you'd like to build a housing estate on it, but you can't because there's TNT in the soil, then the way to get rid of that is to actually go and isolate some soil from the airfield and just get the bacteria out of it, and you'll find that some of those bacteria are actually chomping away on the TNT. And the only reason there's any TNT left is because there are not enough of those bacteria. So you basically just get rid of the TNT by taking these bacteria, expanding them up in the laboratory, putting them back in the environment in larger numbers. And it really works. So bioremediation is not just an academic concept, it's an extremely successful commercial enterprise. And bacteria have been found that break down plastics and rubber and dioxins and you name it. So I realized that there are environments that are enriched in human remains. And of course this seems to be a promising application for this concept. Because if we look at graveyards, then we see that they are indeed accumulating certain parts of the human body, but the ones they're accumulating are the ones that are not energy rich, bones and teeth in other words. Ones that the bacterial ecology would not benefit from being able to break down because they couldn't live off them. The graveyards do not seem to accumulate these nasty chemicals that accumulate in the human body when the heart is still beating. So maybe this is because there are bacteria in the soil that are able to break these things down even though we can't. And I'm not suggesting, in case any of you are nervously worried, that we should inject ourselves with bacteria that can break down oxidized cholesterol or whatever. What I'm suggesting is that we might be able to find the genetic basis for how these bacteria break these compounds down. And having done so, we might be able to take just one or two genes and put those into our cells, thereby augmenting our ability to break things down ourselves and getting rid, of course, of anything that's already accumulated. So graphically, this is the idea. Essentially, there's a process in the human body that turns young people into old people and eventually into dead people. And then there's a completely different process in the microbial ecology of the soil that turns dead people into decomposed people. And all we need to do is figure out how they do it, do some basically standard molecular biology on that process, apply it to our own cells, this is my terrible cartoon of a neuron, and thereby slow down the rate at which the original process happened in the first place. And it turns out it works. So 7-ketocholesterol is public enemy number one in atherosclerosis. It's one of a number of different oxidation derivatives of cholesterol that are found in atherosclerosis, but it's the most toxic and it's also the most abundant. And so we were very keen to try and find bacteria that could break it down. And this is a representative experiment. What we have here is maybe half a dozen different bacterial strains that we give a whole bunch of this stuff to in a petri dish, just like this, and we don't give those bacteria anything else to eat. So if a given bacterial strain can't actually process this stuff, then it just sits there. The bacteria don't grow because they've got no source of energy. But as you can see, that's what happened with most of these strains, and two of them, it didn't happen at all. Two of them were able to metabolize this stuff, and as you can see, in a very short space of time, only 10 days, it was all gone. And this was published last year. This was actually, I think, the first publication that came out of this line of research, and there's a whole bunch more in process right now. So that's all very nice. We need to move on, of course, from there to figure out the genetic basis, to figure out what the genes are, what the enzymes are that are doing this. And there are various approaches to doing that. One approach that we're using in one of the labs that the Sense Foundation is funding is mass spectrometry, which is essentially to identify the breakdown products of the enzymes that are breaking down oxidized cholesterol, and then from there figure out what the enzymes have done, and therefore what the enzymes are, and find them as a result. So that's going quite well too. Now, I do want to make absolutely clear that I don't think this is easy. This is a long shot. It's going to take a long time to get this to work. We've only got to step 2 out of the 7 steps that I have on the slide behind me now. Getting to step 3 is going to be hard enough, modifying the genes so that they still work inside a mammalian cell rather than a bacterial cell. There are various little tricks there to do with compartmentalization, to do with pH, and so on. We think we know how to do those things, but we haven't done them yet. And after that, there's a whole load more stuff to do to see that the things actually work and they don't destroy things we'd like them not to destroy, and then we've got to make them work in mice as well before we can even think about doing clinical trials. So it's a big, big deal. But the fact is that when this works, it's going to be so astronomically more effective against this disease, atherosclerosis, against Alzheimer's disease, against macular degeneration, and other diseases. It's going to be so much more effective than anything we have today, or even anything that's coming down the pike, that the fact is we should get going now. The sooner we start trying to do this, the sooner we really will put those diseases behind us. All right, that is really all I want to say in terms of biology and in terms of what the near-term objectives of the Sense Foundation are over the next couple of decades. What we want to do is develop therapies that clean up the molecular and cellular damage of aging well enough to give middle-aged people maybe 30 additional years of healthy life. But that will not be the end of the story. And I think it's rather important for everyone who's interested in the concept of combating aging to understand what the end of the story is. And it comes down to this concept that I've called longevity escape velocity. The title of my talk, if you remember, was Prospects for Defeating Aging Altogether. And 30 years of life extension, it may be very, very attractive, but it ain't defeating aging altogether. So what's the catch? Well, the big thing is that repairing damage buys time. If you take someone who's, let's say, 60 years old, and they've got 10 years to go before they're going to start exhibiting pathologies of aging, and you slow down the rate of aging by, let's say, 30%, then you're buying those people an extra couple of years of healthy life, which is great. But that's really all you're getting. If you take someone who's 60 and you repair the damage that they have accumulated already, even if you don't repair it completely, if you repair it, like, let's say half of it, but half of each of the various types of damage, then you can realistically expect to give these people maybe 20 or 30 years of additional healthy life. And that's a very big deal. But it's even bigger when you start thinking about what happens next. So I'm showing the concept here very schematically, so don't read too much quantitative into this picture, but I hope to be able to get the general message across. The red line here is the way I'm going to represent normal aging in the absence of any therapies at all. So on the x-axis, of course, we have age, and on the y-axis we have reserve. Now, reserve is not a new concept that I'm bringing in here, it's simply the inverse of damage. So you can think of reserve as the amount of additional damage that someone can afford to accumulate before they start exhibiting pathology. So a person starts off at age 0 with not much damage, lots of reserve, and they gradually accumulate damage, they lose reserve, and eventually they hit this dotted line that I'm calling the frailty threshold, which is where pathologies of aging start to emerge, and not long after that they die. And so the pink line is the, it denotes the stuff I've told you about so far. We take someone who's, let's say, 60 years old, we fix up a lot of their damage, so in other words, we rejuvenate them, and then they continue to age at the same rate that they were ever aging, because remember, we have not altered the rate at which damage is laid down, but then we can bring them in again, and we can rejuvenate them, and we can do it a third time, and so on. And that's why we get a respectable amount of additional healthy life, even if we only fix maybe half of the damage. Now, The problem is that's all we get because the therapies experience diminishing returns. They work less and less well, and we have to do them more and more often. And the reason for that is easy to see on this graph, namely that this is the easy damage that we're able to fix. The difficult damage that the therapies don't work on is also continuing to accumulate, unabated, during the person's life. And eventually, it on its own, however well we get rid of the easy damage, the difficult damage is going to be enough to induce the pathologies of old age. However, the x-axis here, on which I have put no numbers, could be, let's say, 1 year, 2 years, 3 years if we were talking about mice. But since we're talking about humans, it's maybe, 20 years between the first and second rejuvenations of this individual. And 20 years is a long time in technology, a very long time, including, of course, in biomedical technology, which means that it's actually not at all realistic to suppose that the person who comes in here and is rejuvenated will then get this the second time around. What's much more realistic is that the therapies that are applied the second time to this person will actually be more sophisticated, more advanced, more powerful, more comprehensive than the first time. So that's what the orange line denotes, that the person is rejuvenated by therapies that, of course, repair all of the various types of damage that they could repair 20 years previously, but they also repair some proportion, so for sake of argument, half, of the damage that could not be repaired previously, the difficult damage. And as you can see, the long-term implications of that, of taking that into account, taking progress into account, are rather profound. The long-term effect is that we do not see diminishing returns. We actually have these people getting biologically younger as they get chronologically older without limit. People who are receiving the state-of-the-art therapies are as protected from age-related pathologies as they would be if these therapies had been completely perfect from the beginning. Even though, in this scenario, they are actually never perfect. There's always some residual damage that's too difficult for the therapies. And I've given a name to the rate of progress that is required in order to have the general trend be upwards rather than downwards over the long term. I call it longevity escape velocity. If you want to define that in words, it's this horrible long thing, the rate at which rejuvenation therapies must improve following the blah, blah, blah. But as you can see, it's a very simple concept. Now, you'd like to know really whether that's realistic. Is it in fact likely that we would improve biomedical technology fast enough to stay one step ahead of the problem in the manner that I just showed you on that graph? Well, I'm going to give you some evidence that I think that's true. We can start by looking at other technologies. Of course, we don't have any direct evidence because we haven't got anywhere with aging yet. But if we look at other technologies, we can see that the rate of progress we might be able to expect is easily in excess of what's required. So it took a very long time for people to figure out how to fly, but once they did, they were able to push things forward and fly better and better really rather quickly. I bet the Wright brothers didn't think it was only going to be 24 years before someone would fly solo across the Atlantic, and I think Lindbergh would have been quite surprised to see a commercial jetliner just 22 years after he did that, and so on. This is obviously not a unique case. Most technologies behave this way. We can think about computers, for example. We can think about the combating of infectious diseases. When Jenner and Pasteur were figuring out the very elementary aspects of how to combat diseases, they were able to save a bunch of lives. But once they'd got that far, which was of course much further than anyone had got before, progress was relatively smooth and relatively rapid. So I think it's pretty reasonable. In order to reinforce that even further, a couple of years ago I did a computer simulation of the aging process, represented in this proper way with metabolism causing damage and damage eventually causing pathology. And to cut a long story short, the simulation reinforced this concept very powerfully. What we have here in the black line on the left-hand side is the survival curve of a cohort of humans who are not receiving any therapies. So a survival curve is simply a plot of how many people are alive from an initial population at any particular age. So of course at age 0 everyone's alive by definition, And thereafter, not many people die until age 50 or 60, then they start dropping like flies, and not many of them get beyond 100. Now the blue line, the dark blue line right next to it here, is the trajectory, the survival curve of people who do receive these therapies, but who only start to receive them starting from the age of 80. Okay? And as you can see, not a lot happens early on. In fact, of course, at first nothing happens at all because the therapies aren't yet being applied. Half the people are dead by the age of 80, thereabouts. And then after that, people get a little bit of benefit, but only a little bit, because these people are frail. They are biologically 80-ish, and they have a high chance of death each year, so just none of them really get through. But look at the next line. Look at this line here, the grey-blue line. That is the survival curve of a group of people who were just 10 years younger, 70 years old at the time that the therapies were first applied. And it's the same story at first, of course, but eventually people start to get really seriously biologically younger as they're getting chronologically older, and the risk of death from age-related causes goes down and down and down. And by the time we get to 150, 15% or so of them are still alive, and the chance of death from aging is pretty low. And when we get out to around 200, it's become essentially zero, so that about 10% of these people actually make it to an indefinite age, such that they're only dying of things like being hit by trucks, things that currently affect young adults. And if we carry on this, this concept, we can see in the brown line, for example, which is the people who are age 60 when the therapies are first applied, nearly half of them survive. So this is all rather splendid. This is longevity escape velocity in action, and it's even better than that, because as you will see at the top of the slide, I made very, very pessimistic assumptions in this simulation. 42 years, I mean, that's the time between Lindbergh and Concorde. So suggesting that the therapies would only double in their efficacy in that time is just, insanely over-pessimistic. So I really want to point that out, because it's insanely over-pessimistic, and still it was enough to give this longevity escape velocity in spades. Cars are a fine example of this. Again, of course, I showed you some cars earlier on that were 50-odd years old. This car's more than 100 years old, and it's doing just as fine as it was when it was first built. And the best of it is, okay, yes, it's getting a lot of maintenance, but it's not getting any more maintenance than what it was, let's say, 30 years ago, when it was only 3 or 4 times as old as it was designed to be. In other words, longevity escape velocity for cars is already more or less zero. We hardly have to do any improvement anymore. So what this means in terms of lifespan is interesting. I want everyone to understand here that I do not work on getting people to live a long time. I work on keeping people healthy. It's just that I think I know how to do it really, really well. So well that there will be this side benefit that people will tend not to die very often. But what it means is that the hard part of defeating aging is the first part. As most of you probably know, the world record lifespan at the moment is 122. A splendid lady named Jeanne Calment, who died in south of France about 12 years ago, and who said on her 120th birthday, when asked the inevitable question, what is your secret? She said, my secret is I gave up smoking when I was 117. So in order to get to 150, we're going to have to actually do this robust human rejuvenation, this additional 30 years. And once we've done that, the hard part will be over. And people who are not much younger, maybe 10 years younger, certainly I think 20 at max, will be able to escape aging indefinitely. And those people will have a good chance of making it to 4-digit lifespans. Because if we do the arithmetic now, if we ask, what are the risks of death in young adulthood in the Western world, we find that if those risks were perpetuated and not increased with age throughout subsequent life, then 1,000 years is the sort of average lifespan that we could expect to see. What it doesn't mean is this. This is the sort of thing that I get as knee-jerk reactions to what I'm talking about. Immortality is the purview of religion, it's not technology. This longevity escape velocity thing is obviously, roughly speaking, immortality. And since all of these technologies, these rejuvenation technologies I've spoken about, are apparently implying longevity escape velocity, then we can add all this up and we can say, well, these technologies are science fiction. Now, of course, that's ridiculous, But it is the sort of thing that I get lambasted by sometimes, even by people who ought to know better. Tom Kirkwood and Richard Farragher are two of my good friends. They are very prominent professional gerontologists, and they say things like this about my work. But the fact is, I think it's they who are irresponsible. I think that such reactions are slowing down the enthusiasm for the regenerative medicine approach to combating aging, and therefore actually costing lives. So when it comes to that, the question is, how can you make a difference? One thing you could do is get hold of this book, and I prostrate myself at this point because the plan was that I would actually have like 50 of my book here for you to take off my hands at a respectable price. And I forgot to bring them. They're in the house of a colleague of mine in Redwood Shores. But I'm hoping that I can get them down here so that they'll be available for people to buy when you next meet in a month from now. I'll try and arrange that with my good friends over on the left. But you can, of course, look at our website as well. You can send us an email. And so I hope that will all be all right. Oh, before I forget, actually, I do actually need to go back to Redwood Shores sometime today. So if anyone happens to live there, oh, you do you? Okay, great. Thanks very much. Well volunteered. Finally, I just wanted to mention the foundation. We are obviously a charitable foundation. We support a lot of science, and we do it with money that other people give us. So it's important to know that if you do feel like any sort of financial contribution, it will be very well used. However, I also want to emphasize on this slide that we don't just need money, we also need people to volunteer their time. There are plenty of things that we would like done that we can't afford to pay people to do. And so, as I say, these audacious ideas can be made into a reality, perhaps with your help. And you're always welcome to write to us and see what we can do with you. And I'll stop there. Thank you very much. Yeah. I really should have it on the last slide as well, shouldn't I?
I had a question for you. It seems like you made the assumption that the proper delivery mechanism for these cool substances made by bacteria that live in dead people is genetic. Isn't that kind of a lot of busy work? Couldn't we just figure out what the enzymes are, like with nattokinase, put them in a pill and take them?
Okay, so there are, maybe is the short answer. These enzymes need to get to the cells in which the stuff that we want them to break down are accumulating. And some of those cells are not so easy to get to as others.
DMSO maybe?
Well, yeah, I so much DMSO I'd want to ingest, I think. So gene therapy may be the way to go, depending on how good we are at gene therapy by the time we've identified and suitably modified these enzymes. However, of course, there is the option of enzyme therapy, which does not mean ingesting these things, it means injecting them, but still it means doing it without actually messing around with genes. And this is already used as a very successful approach to treating a family of diseases called lysosomal storage diseases, which result from the congenital absence of a particular enzyme that normally breaks things down inside you and me. These diseases are vicious. They are, by and large, childhood fatal because some stuff or other accumulates in the cell that would normally be broken down. And the way they are treated is by the creation in the lab of a lot of this enzyme and the injection of heroic amounts of it into the bloodstream. Actually, there's small modifications made, glycosylation changes, so that the enzymes are, at least to some extent, picked up by the cells that need them. But these therapies really work. There are people walking around as adults, having perfectly normal lives and families and everything, who would have died at the age of 5 if it hadn't been for these therapies. So that may very well be, at least in the case of some of the diseases that we want to address, it may be the mode of delivery of choice. Yeah?
In terms of your model of the escape velocity, if the aging after the rejuvenation is slightly faster, how does that affect your projections?
Well,
Instead of it being perfectly
Well, right, yeah. So this is sort of why I didn't, why I emphasised that one should not really take anything quantitative out of this, because I was trying just to get this concept across. Essentially, I've got what you're saying on this slide already, because I said that I expected us to get maybe 30 years out of the first generation therapies, which is maybe an extension of 30 or 40% of life, and you see what this looks like is almost a doubling of life. So actually, if you think of it, if you think about the bottom half of the graph being bent round a bit, so that the red line really does indeed do this, accelerates, then you'd see the other things accelerating as well, and you'd get more or less exactly what you're talking about. So the simulation that we did in here did indeed do exactly what you're saying. It did indeed incorporate the fact that we would expect an acceleration of aging at greater chronological ages, which would have to be compensated for by progressively more effective therapies. Yeah.
Is there a branch of sense that's looking at the, I don't know, the sociology of super longevity?
Well, sort of. Yeah, we did actually, we're definitely interested in this, not least because we need to reassure people with purse strings that the sociological consequences are not reasons not to go this way in order that they'll give us their money. So yes, it is an important thing. And one thing, probably the most important thing, is the question of what it would do to population. People worry enormous amounts about this. We actually funded a couple of demographers in Chicago last year to do the first stage of a very thorough study on exactly this, on, well, basically to write some new software to do projections of what we would expect for global population in the context of various assumptions about when the therapies come along, how rapidly they are disseminated, what happens to the birth rate as a consequence, all those things. So yes, we're definitely interested in all of that. Yeah.
You've been showing these rejuvenations as discrete events rather than continuous processes, and I'm curious why you do that, or is it a continuous thing that's really dermatology, or how
yeah, so you're quite right. I mean, here I'm really just trying to emphasize the improvement in the technologies, but you're absolutely right. Some of these therapies early on may be rather arduous, like may involve surgery, for example, and so you wouldn't want to do them more than once every 10 years or so. But as time goes on, and especially as the therapies not only become more comprehensive, but also more convenient and cheaper and so on, they may get to the stage of being outpatient jobs or even self-administered things. And at that point, you can definitely look at options that one can take just as frequently as one currently takes a supplement. You're quite right. Yeah?
It seems to me that you're oversimplifying in a very serious way. Like, for example, you gave this example of getting rid of the certain bad cholesterol.
Yep.
If you could do something that inject somebody with something where all that would be fixed in an hour, there'd be 100,000 other problems that would now be there just below that that are going to still be there. And you have to, it seems to me you have to do a complete draw assault on a whole bunch of things to get the kind of dramatic effect.
Okay, so that's really what this sort of stuff comes down to. So let's take that particular example, because it's easier to do it with a concrete example.
You're showing about 5 or 6 things there when there may be hundreds of thousands.
That's right. So, okay. So what I'm saying is that the categorization that I showed you earlier on into 7 major types of damage. Okay. We're not going to actually achieve robust human rejuvenation without fixing all of those types of damage fairly well. But then we have to ask, well, what does fairly well mean? So let me use your specific example. Suppose you could get rid of all of the 7-ketocholesterol. You'd still have other types of oxysterol, for example, accumulating in the lysosomes of macrophages in the arterial wall. The thing is that those things are additive. It doesn't matter which oxysterol there is. They all poison the cell, the macrophage, in the same way and turn it into a foam cell. So if you've got, let's say, 10 different oxysterols, okay, but one of them happens to be present at a level equal to or greater than the sum of all the others, then if you just fix that one, you've halved the problem. And you've bought yourself time to develop the fix for the second one, which will buy yourself time to...
You need to pick the most important and the next most important and the next most important. Is
Well, at least, yes, at least that would be the ideal order of events. That's right. Yes, but certainly the.
I hope that the ratio from the first to the second to the third is dramatic.
Oh yes, oh yes. Well, actually, we're lucky. It is. It always is. So, in the case of, for example, glycation, of cross-linking in extracellular collagen, again, same deal. There's one particular cross-link called glucosapane, which is far more abundant than all the others put together. And so, yes, this is generally the case.
But so, all right. So you got an 80-20. You get rid of the 80. Now, of those 20, is there another 80?
Oh, absolutely.
Or is it
Absolutely.
closely packed in?
To the best of our knowledge, it's not closely packed at any point. And also, even this doesn't apply in most cases. Well, so it applies for junk. It applies for extracellular junk like amyloid as well. And this is just one trick, and the only thing that will be progressively improved is the efficiency of delivery of this. So you may be able to have gene therapy that only gets to half the cells the first time, but you improve the vectors so that the next time you can get to 90% of the cells and so on. You get the idea.
So does your book actually discuss many of the therapies that you think are appropriate?
Oh, absolutely. That's what the book is about. Yeah, absolutely. The book is 90% science. And each of these 7 categories is the subject of at least one chapter of 20-odd pages of details of where we are, what we need to do, how we're going to do it. Yeah, absolutely. That's what the book's about. And I should emphasize the book is written for a general audience. It doesn't cut any corners with the science. It's probably at the level of technical detail of A Brief History of Time, for example. So it definitely is written for a general audience, but you're probably going to have to take it slowly. Yeah.
As you discussed this theory, I was just hearing the progress the science has done probably over the last 100 years. I mean, 100 years ago, most people would die within very young age, and vaccines and that. So every one of those incremental processes have taken with what he was suggesting. I mean, now you have the damage is so entangled and so complex that it's no longer going to allow you to.
So I understand your caution, but you have to look at the details in order to know whether that's really true. So, I mean, yes, it was much easier to fix the problem of infant mortality more or less almost completely than it is to fix the problem of aging. But that's why we've already fixed the problem of infant mortality and we haven't fixed the problem of aging yet. As progress goes on, we'll get to a point where we can cope with the complexity of aging, and the essence of my work is to tell you that I think we're nearly there. It's just a question of how soon we get there. And I don't know that we're going to get there in 20 or 30 years, but I think we have a respectable chance. And the reason I think we have a respectable chance is because I can describe the details of what we need to do. Yeah.
Do you give any credence to the peptide exchange between organic carbon versus non-organic filtration systems?
No.
It slows down the ethylene decomposition of lead.
I don't know anything about that at all, no. Yep.
Don't you have the war? I mean, I'm listening to what you're talking about, the microbes and wiping out all the nano, whatever it might be. But don't you have the basic problem of man and how he behaves and what he adjusts and how he thinks and all these kind of things to counteract it?
Not really, no. So, of course, there is a difference between different people in how fast aging happens, and also within the same person in how fast different aspects of aging happen. And some of that is lifestyle driven, some of it's genetic and so on. But the great thing about it is that the variation is actually relatively small. A factor of 2 maximum between the fastest aging people and the slowest aging people. Now, that's great because these rejuvenation therapies, these repair and maintenance therapies, will get rid of damage much, much faster than it's laid down. Even for faster aging people, aging is really slow compared to the rate at which these therapies will reverse the aging process. So yes, people who are living punishing lifestyles or who are genetically unfortunate or whatever may need to benefit from these therapies more frequently than people who are luckier or who are more conscientious, but they'll still be pretty much the same therapies across everybody. And that's of course rather useful because personalized therapies are more expensive than generic ones. Yep?
I hope you will consider not using animals in your continual research, using other methods, cell lines or computer-aided design.
Oh yeah, I mean, of course, we take the same view of of animal research that most biologists take. In other words, we only use them when we absolutely have to. But of course, the same logic applies as for any medicine, that ultimately you've got to decide how close to get to the human being before you go to human beings themselves, because you have there's a trade-off between how many humans you want to kill with your experimental therapies. Any more? Yep, in the middle.
May I ask how old you are?
Yes, you may. I'm 45. No, I'm not. I'm 46. Curses. I'm old enough that I have to think about it. Right. Any more? Yeah.
So back to your first chart going from metabolism to damage
Yeah.
to pathology. I personally experienced, and I know other people have experienced, that you can, it is a huge intervention between metabolism and damage. You just clean up your act and not eat too much and eat only nutrient-dense foods.
Yeah.
And you have to say anything about that.
Yeah, I mean, I did say a little bit about how I think it's important not to overemphasize that. Now, I do agree that one can make a difference, and of course, for some people, it can make more difference than for other people. So, for example, I know Ray Kurzweil pretty well. Now, he takes an insane number of supplements. Well, I say it's insane. I don't think it's an insane.
I'm talking about supplements.
Wait, no, I'm just using it as an example. Just an illustration. He takes a very large number of supplements. I would never dream of taking all of those supplements, but for him, he came down with type 2 diabetes in his 30s, so he's unusually unlucky, and he developed this whole scheme of his, essentially starting with trying to solve that problem. Now, I think all of the various simple things that we can do, including the ones you just mentioned, have the same sort of situation. They can be considerably beneficial for some people, but I think that at the moment, the overwhelming evidence tells us that for people who are already reasonably well built and are going to live to the age of 80 in reasonably good health anyway, nothing exists at the moment, whether it's lifestyle, whether it's supplements, whether it's any of these things that you're talking about, that can add more than a year or two to that.
Oh, no. Cutting back on calories to your optimal level is easily a 20, 30-year improvement.
I don't think so. And I'm telling you why I don't think so, because there's anecdotal evidence already. So, of course, Okinawa is the best example of this. Okinawa is an island, as many of you know, at the south side of Japan, in which the genetics is more or less the same as mainland Japan, and the diet is more or less the same in terms of the composition, but they don't eat so much. Or rather, they used not to eat so much until they discovered McDonald's. And sure enough, they live longer than mainland Japanese. But how much longer? 2 years. So it's important not to overstate this. And that's just one, that's just the best example, but there's plenty more examples. Calorie restriction in mice and rats gives maybe 30 or 40% additional life, but only if it started very early in life. Okay, if it started in mouse middle age, then you only get 5 or 10%. No surprise. See? That's right. Exactly. And but the worst of it is that, as I mentioned earlier, there's good evolutionary reasons, and actually I think good data, to suggest that for longer-lived organisms, you won't get so much benefit from dietary manipulation anyway. Essentially because dietary manipulation, especially manipulation of dietary calorie intake, is something that the body knows how to respond to because it has been useful in the real world to be able to get through a famine. And famines that are a year long happen often enough for evolution to pay attention, whereas famines that last 20 years don't happen often enough. So in other words, we've lost the ability to respond so impressively as shorter-lived organisms do, in the same way that we lost the ability to make our own vitamin C.
Except that we're not really talking about famine, we're talking about gross overindulgence versus reasonable.
Oh, well, don't get me wrong. I'm certainly not, remember, I come from a country that isn't nearly quite so addicted to food as you guys. So perhaps I misunderstood your question. Absolutely, it's very easy to shorten one's life. And one can shorten it very considerably by eating the sort of way that quite a lot of Americans do these days. I wasn't denying that at all. Yeah?
What happens if there's another mechanism of aging that you're not considering? What happens if there's a mechanism involving the stability of the genetic networks that underlie cellular metabolism, so that, for example, small variations in poorly engineered networks that were not selected to be running for more than a certain number of cell divisions for a certain amount of time fail.
Yeah.
Obviously you can engineer it once you understand it, but that brings back the whole problem of understanding the calculus.
Right, absolutely. Yes, if such networks existed, then I would be worried for exactly the reason you just described. We would have to understand the network very well in order to manipulate it. However, even though I can accept that in theory such networks could exist, such sort of metastable networks, if you like, in practice, no one's ever found one. And that's not surprising. You expect a lot of stability. You expect a very powerful homeostasis in any organ.
I would say that there actually are stable networks that characterize, I mean, there are genetic networks that are characterized by various epigenetic effects.
Sure,
See,
but they're stable.
Some of them are actually running on very few molecules. So there are known cases in the biomedical literature where you can just do a switch transition from one state to another based on just changing a little bit of the expression of one protein. I can give you some examples.
Sure, I know that's true. But you're talking about, but your question was not about the stability of, or the particular level of the sensitivity to the level of one particular protein. Your question was about the stability of the overall network.
Well, and that affects the stability of the network.
That's what I don't think there are examples of.
I think I can show you. I can email you.
Do that. I've worn you out. Excellent. Thanks very much. Thank you. Thank you. It's been a real pleasure. Well, I enjoyed it myself enormously.